Assalamualaikum. My name is Ikra Aftab, and the topic that we're going to present is testing of COVID-19 and other viruses in the laboratory. We will see the chemistry behind the diagnostic kits. So first, we should know what is COVID-19. It was a viral disease that was spread in different parts of the world, also known as coronavirus. Instead of this virus, we will also study other viruses that are being tested in the laboratory. So the main objective is to study the different test kits that are used for the detection of COVID-19 and other viral diseases. We will study different test kits that have been used to detect these kind of viral diseases. We'll understand the basic theory, the chemistry, simple step-by-step methods, reasons, their uses, and we'll see that how we can improve them in future. Let's go to the basic introduction that what are viruses? Viruses are germs that can cause diseases in humans, not only in humans but also in animals. So testing for these viruses like COVID-19, HIV, dengue and hepatitis, it is very important to test these viruses so that we can control their spread, because if they spread more, the population will decrease a lot, which will affect the economy. Different test kits like RT-PCR, rapid antigen test and ELISA are used in the labs. Each method of testing is different, but they uses the basic chemistry principles and biology for detecting these viruses. So all this basic chemistry we will see in the theory that is explained further. So what is the theory and chemistry behind the test? RT-PCR, reverse transcriptase polymerase chain reaction. It is a test that finds that RNA is of a virus like COVID-19. COVID-19 is a virus known as corona, so this test is used to find the RNA of that virus so that it can change that RNA into DNA and make so many copies easily. So when RNA is changed into DNA and many copies are made, it can be seen easily. The chemistry behind this test is that it uses the enzyme which is called reverse transcriptase. This enzyme, reverse transcriptase, helps to change RNA to DNA. Then the DNA polymerase enzyme makes copies of the DNA and we can see them easily. Special chemicals are also used like primers. These chemicals help in copying the right part of the virus. We can also use a fluorescent dye or probes which gives light when copies are made. So when the light is shown, it shows that the virus is present. Just like a proof kind of thing is here, light acts as a proof that shows that our virus is present. This was the first chain reaction, RT-PCR first test, and we have seen the theory, we have seen the chemistry. The main thing is that you have to change RNA into DNA by using different enzymes, and then you have to make the copies of that DNA. The second one is rapid antigen detection test. This is a very quick test and it finds the proteins in the virus. Now in this virus, if you have a virus and you have to find the proteins in the virus, you will use the rapid antigen detection test. The test strip has antibodies that can catch virus proteins. This test has a strip and the strip has the antibodies that helps to catch the proteins that are present in the virus. They're connected to colored beads. When the virus proteins and antibodies they join, what happens is that they make a line on the strip. When a line on the strip is made, it shows that the result is positive, or you can say it shows that the virus is present. So rapid antigen detection test is simple, the chemistry and theory is simple. Let's go to the next test, which is the ELISA, enzyme linked immunosorbent assay. ELISA is used to find antibodies or antigens in the blood. So if you want to find antibodies or antigens in the blood, you will use the test known as ELISA. What is the chemistry? What is the theory? A plate has antigens or antibodies stuck onto it. If the patient sample has matching antibodies, then the antibodies present on the plate and the antibodies of the patient, they join together. Then another enzyme linked antibody is added. A chemical TPMB is also added, which changes the color. If the enzyme is present, the color is checked using the machine. So if the enzyme is present, the color is changed. And how will you know that the color is changed? You will check in the machine. In this test, different materials are used like sample swabs, RT-PCR chemicals, which includes buffers, enzymes, primers, rapid test strips, ELISA kits, pipettes, tips, gloves, masks, PPE, spectrophotometer, PCR machines, and waste container. What is the process of this RT-PCR test? What is the process of rapid antigen test? What is the process of ELISA test? Let's look at these procedures. In RT-PCR test, you have to collect the sample using a swab. Then you have to mix it with lysis buffer so that the virus breaks and release the RNA. Then you have to use the column to collect that RNA so that RNA sticks to the silica. Then you have to add the reverse transcriptase, which is an enzyme, so that it can change our RNA into DNA. Then you have to add DNA polymerase enzyme, primers, and dye so that many copies of DNA can be made. Then you'll run the machine and check the light signals. If there is more light, it means more virus is present. The second one is rapid antigen test. Collect the sample with the swab, mix with the extraction buffer so that it releases the protein. When the protein is released, you have to put the drops on the test strip. When you put the drops on the test strips, the protein moves and reacts. Wait for lines to appear. When the lines appears on the strip, it means that the virus is present. ELISA test. You have to add the blood sample to the plate, wait for a while, and then wash it. It keeps only masked antibodies. Then you have to add enzyme linked antibody, wait and wash. Then you have to add TMB chemical. If the color shows positive, add acid to stop the reaction. Check color using the machine. Color strength shows how much amount of virus is present. Now what you are observing in RT-PCR, you are observing the light signal. If the light signal increases, it's a positive test, means virus is present. In rapid test, you see the line. If line appears on the strip, the test is positive, virus is present. In ELISA test, color appears. If color appears and strength of the color tells how much amount of virus is present, but if the color appears, the virus is present, the result is positive. Positive means the virus is present, and negative means the no signal, no line, no color, no virus. It has various applications. It is used to detect COVID-19, also known as coronavirus, HIV, dengue, hepatitis, screen people in outbreaks. It is also used to test patients in the hospitals. It is also used to check blood before transfusion. So these tests have many applications. 4 of them are given here. What is the future recommendations of these tests? They will help to make the process faster. They have portable kits. You can carry them anywhere. You can use AI for automatic results. Use CRISPR for very accurate results. Make kits for many viruses at once. Make cheap, easy kits for villages, so that the people that are present in villages can also do these tests. You have to make the kit so that many viruses can be detected at once. But make sure you should wear gloves before doing the test. You should wear mask. Use new tips for each sample because the dirt can affect the results. Throw waste properly. Keep kits at correct temperature. Check expiry dates of the kit. These are the precautions that must be kept in mind, but these tests are very beneficial for detecting the viruses that are viral viruses, means they're spreading. Viral means they spread from one person to other. So all these tests are very important. We have seen the procedure, we have seen their application, we have seen the chemistry and everything behind it. That's all for today. Thank you.